Good morning, everyone, and welcome to our network. It's my pleasure to present to you guys Dr. Luger Stanker. I think that's the correct pronunciation.
Yes.
Luger Stanker. He is from the university, from the Ulm University in Germany. Is that correct also?
Ulm, con U.
Ulm. The Ulm University in Germany.
Yes, yes.
Dr. Stanker is a member of the investigation group named Peptide Libraries Purification, Synthesis, and Structural Analysis. And he is also a very close collaborator with Dr. Anselmo in Cuba. We didn't get his curriculum vitae, but from what we've seen online, he is a leader in antimicrobial peptides, and also antiviral peptides. And he has a lot of important work with antiviral peptides, especially against HIV/AIDS.
Yes.
So without further ado, I hand the mic over to our invited guests.
Okay, thank you very much, dear ladies and gentlemen. Thank you, Professor Carlos Munoz, for this seminar invitation. It's a pleasure for me to present you our group, our research work, and our technical possibilities, which later might serve as a basis for future collaborations. So I'm heading the core facility of functional peptidomics, which belongs to the medical faculty of the University of Ulm. It exists since 2017, but there was a precursor which was named UPEP, Ulm Peptide Pharmaceuticals. It was the competence centers for peptides existing in Ulm since the year 2012. And since 2020, we have moved, as you can see in the picture, to new laboratories in the Central University of Ulm. And you can find information about our service, about our research on our homepage. Just give in core facility Ulm University and you will easily find us. So from my curriculum, I myself was working for more than 20 years in Hanover, in Germany, in a peptide research center, which was founded by Professor Forsman. And I will later give you some more background information about this time, what we developed there in Hanover. And then we transported this experience to Ulm University. So our group is quite small. We are only 4 fixed persons, 2 technicians, and myself and Armando Rodriguez, who was graduated in Havana University. The city of Ulm is a quite small city. It has only 130,000 inhabitants, but it's a very potent city. It has a university with more than 10,000 students, and it has a technical university with more than 4,000 students. And it is close to the city of New Ulm, which belongs to Bavaria, which is on the other side of the Danube River. And altogether, they are living in the big area of Ulm, 200,000 people. And the most important thing in Ulm is, of course, the Ulmer Münster, because it has, they say it has the biggest church tower of the world. So it's quite famous and there's a lot of tourism in Ulm. It's located between Stuttgart and Munich in the south of Germany. So the hypothesis we developed together with Professor Forsman around 30 years ago was that from abundant plasma proteins, a high number of yet unknown regulatory peptides are released and to be discovered. So very briefly, what is a peptide? A small protein with less than 100 amino acids. Generally, peptides are cleavage products of such proteins, or, for example, in the case of chemokines, they are specifically released on a stimulus. And on the other side, we often see that the cleavage of proteins is very specific in response to a physiological or pathophysiological stimulus to generate bioactive peptides. So I think in the future, peptides will play a much major role on the pharmaceutical market because they have already a proven history. As you can see in this table, insulin is used since the 1920 years against or for treatment of diabetes. Adenocorticotropin is used since the 1950s. Calcitonin is used since the 1970s. And there are several blockbusters on the market, like leuprolin, used in prostata cancer. And I think peptides will have a very promising future as therapeutics. And at the moment, there are more than 150 peptides in clinical development. For example, you can see, okay, this is the status of 2018. You can see 25 peptides in phase 3, 75 peptides in phase 2, and 50 peptides in phase 1 clinical study, which I think is a lot. And also quite interesting, one aspect, I think it's more useful to use smaller peptides as therapeutics, and the tendency is to use peptides in the range between 2 and 10 amino acids, which is the orange bar in each of these diagrams. And of course, because the synthesis of smaller peptides, it's much cheaper, and the bioavailability in most cases will be much more easy. So what are the molecular targets of pharmaceutical peptides? So the most important target are the GPCRs, as also common for small molecule pharmaceutics, but also catalytic receptors or ion channels. Only a small portions at the moment of the peptides that are launched as drugs or developed as drugs are used as antimicrobial targets. But hopefully this also will increase in the future. As you may know, there are quite huge challenges also, because usually peptides have a very short half-life because they are cleaved by proteases. There's an excretory clearance via the kidney, and without any modification, there's no oral bioavailability because there are digestive enzymes. The pH in the stomach is too low, and the intestinal permeability is quite low due to the relative high molecular weight and the polarity of peptides. But all these points are actually addressed by the research to get the most peptides orally available. So what is our general concept is? Our general concept is to start with a natural source, and from this natural source we create a so-called peptide library, which contains several thousand different natural peptides. Then we start screening, isolation, and identification of the peptides. When we have the sequence of a new peptide, it will be resynthesized. Its activity has to be revalidated, and normally the sequence will be optimized by structure activity relationship analysis. And the final goal is to get a lead candidate, to start a preclinical development. And of course, a drug candidate, but this will then be given to companies. So what we can provide in Ulm at our center, functional peptidomics, we have a lot of chromatographic systems, large-scale chromatographic systems. So we produce extracts, we produce peptide libraries, and another very important work is to synthesize peptides. Of course, this is possible up to a length of around 50 amino acid residues. And to get optimized peptides, we use, for example, modifications like acetylation, amidation, dimerization, or coupling of fatty acids, D and beta amino acids, or for instance, for research purposes, fluorescence labeling. So we have a peptide synthesizer based on microwave technology, which is more rapid than the conventional peptide synthesizers. So since several years, we are focusing on human peptides. So we are extracting peptides from different organs and body fluids. Normally, these peptides, or if in the case of an organ like placenta that we get from the clinic here, the tissue is homogenized using acetic acid extraction, and then it is centrifuged and filtered. And what we normally do is we pass an ultrafiltration step using membranes with a molecular cutoff of 30,000, because we really focus on peptides smaller than 30,000. We don't want to work with proteins because our focus are the peptides. What is very important for us at the moment is a so-called collaborative research center, 1279. Maybe you know the German funding organization, SFB, DFG. DFG is the organization. It's a central German funding organization for research. And we have really a huge funding from them. And so we are a core center within this CRC. You can only also find it on the homepage of the CRC 1279. And in blue, you can see all the peptide libraries that we have processed for this research center which consists of 20 different research groups, mainly at Ulm University. So all these groups, or many groups, use our peptide libraries to screen for antimicrobial, antiviral, or anti-cancer peptides. So the general concept of this research center is that we expect from 25,000 different genes in the human body, more or less 80,000 different proteins are expressed. And from these 80,000 proteins, we expect the existence of more than 1 million different human peptides. Of course, most of these peptides are waste products and will go into the normal protein turnover of the body. But we found in the last decades that from large precursor proteins, for example, from albumin, from fibrinogen, from hemoglobin, small bioactive peptides are released which have completely new and unexpected functions, like endorphins, antimicrobial activity, or peptides as response of inflammation or cancer. This is a general concept of the research center, but as mentioned, it is exclusively focused on human peptides to explore the human peptidome for functional peptides, not to generate sequences, but to find new bioactive peptides. So the story began 30 years ago together with Professor Forssmann in Hanover, because he had the idea that human hemofiltrate, which is obtained from patients with chronic renal failure, contains the most abundant number of different peptides in the human body. So he started, together with me and other co-workers, to collect human human hemofiltrate in amounts of several 10,000 liters. So this was a logistical question, but the hemofiltrate has a big advantage. It contains only small molecules below 20,000 kilodalton. So it was an enriched source of peptides. And you can see our installations in Hanover. This was production in large scale, and this was really a quite successful project. So the process was standardized, and for example, from 10,000 liters of hemofiltrate, first we applied it to a cation exchange column. Later, each of this cation exchange fraction was applied to a reverse phase column and yielded in a total of 300 to 500 fractions. And you see here on the right figure, these are aliquot boxes that are provided to our collaborators, which can use them for standardized bioscreening in 96 well format. So this was a procedure that was optimized during several years in Hanover, and starting with cation exchange, then high resolution reversed phase, and from the peptide fractions, we put the peptide fractions into an high throughput screening assay. Then we see there's an activity in a certain fraction. This hit fraction is then further purified on using chromatographic columns until we have the single purified bioactive peptide. So what is most important in isolation of in discovery of peptides is a very strong analytic. So here in Ulm, we have another core facility. It's the core facility of mass spec and proteomics. And they have a quite good equipment, for example, to clarify the purity of a purified peptide by capillary zone electrophoresis. And what is also most important, mass spectrometry. So here in Ulm, we have MALDI. Okay, we applied now recently to get an own MALDI here, because this is very important, because you can measure mixtures of different peptides very easy and very rapidly. You get the mass information. Maybe you will know this. Usually you see the double charged ions and the single charged ions, and also often you see the dimers of the single charged ions. So MALDI, mass spectrometry, and of course electrospray mass spectrometry are very important for us to get the exact molecular mass information of a peptide. And of course, nowadays, MS is also used for sequence determination of proteins and of peptides. Sometimes we use Edman degradation still, which is useful in some cases, but we have to use a service company for Edman. Here in Ulm, we can make MS sequence analysis. So in this research center, our unit is a central project with more than 20 collaboration partners. And as you can see here, for example, this was the outcome, what we have isolated in new human peptides during the first funding period, which started in 2018. Or yes, I think in 2018. So we found around 20 by bioscreening approach, around 20 new bioactive peptides, most of them fragments of larger plasma proteins. And most of them will be published or are already published. You can see in the fourth line, we have recently isolated alpha-1 antitrypsin, which is an inhibitor of SARS-CoV-2, of COVID-19. There is a pre-publication online. This was quite interesting because alpha-1 antitrypsin is already launched medicament in patients which suffer from alpha-1 antitrypsin, especially in the lung. So it was already used in the clinic by Chinese doctors as a treatment option. Okay, this was an interesting finding. Unfortunately, we have not been the first that found alpha-1 antitrypsin to be an anti-COVID agent. Okay, but we found several new antimicrobial peptides, toxin inhibitors, anti-cancer peptides. So at the moment, we are applying for the second funding period, which will start in beginning of 21. So this is very important, especially for our unit, because 50% of our budget comes from this CRC 1279. Okay, as you may know, nowadays, if you find a native peptide and you want to optimize it, especially in the case of antiviral or antimicrobial peptides, you can just take the sequence and use programs that are available for in silico optimization. And I think this is a quite promising approach. And we were able, for example, the Pomacea 1 was also found in a group of peptides that we have isolated from the Pomacea snail using these programs. So it was not still not optimized, but it was found by such bioinformatic tools. So I think this is quite important also in the future. And just to show you the optimization of the peptides, for example, was quite successful in the case of a peptide. The peptide is named EPX4. You find a lot of publications about it. It's a peptide fragment from human albumin. And it's a very potent inhibitor of the CXCR4 of the HIV co-receptor. And this albumin fragment originally had 16 amino acid residues, and during several optimization rounds, we could reach a much more potent derivative, which is now in clinical development. And the IC50, for example, in the anti-HIV assay is around 1 nanomolar. So from 515 micromolar, the potency could be optimized to 1 nanomolar. So usually the criteria for optimization are the length. Of course, if a peptide is shorter, it's much more convenient. The potency, the stability in plasma, and of course the toxicity. We also have some toxicity models here in Ulm, like a zebrafish model, which can be easily applied and is available for even basic research projects on the basis of scientific collaboration. And in the picture of this slide, you see the founding professors of our facility, and especially I will mention Professor Forssmann, Professor Jan Münch, Professor Frank Kirchhoff from the molecular virology here in Ulm, and Frank Rosenau, who is the expert in antifungal research here in Ulm. We also have a very close collaboration with the medicinal microbiology department, which is Steffen Stenger and Barbara Spellerberg. So as a proof of concept, together with Professor Forssmann in Heidelberg and Hannover Medical School, we have developed the concept, and we have isolated several hundred new bioactive peptides, and 2 have entered clinical phases. One is the ularitide, which is a special form of urodilatin, which is a peptide hormone, and a peptide found here together with virologists in Ulm, VIR 578, which is an HIV fusion inhibitor. But this project will not followed up because there are too many drugs on the market for treatment of HIV, and the treatment is quite sufficient at the moment, we think. So this product will not be competitive. And as mentioned before, the EPX4 at the moment is in preclinical development as anti-inflammatory or anti-cancer compound. So we were broadly screening for antiviral peptides, but interestingly, screening for antiviral peptides, we found in fractions obtained from human seminal plasma, from human semen, a fraction that dramatically enhanced the infection of HIV. And this was an interesting finding. And we found that this peptide, which is a fragment of prostatic acid phosphatase with a molecular weight of 4,500, classical peptide, that this peptide in vivo forms fibrils, like beta amyloid fibrils. And these fibrils, you see here in the picture in green, they attract the virus and function as a carrier of the virus and facilitate the entry of the virus in the human cells. So it seems that this peptide in vivo promotes HIV infection in human semen. So this was physiologically a very important and interesting finding. And it was, of course, surprisingly, because we were looking for anti-HIV compounds, and we found a compound that enhanced HIV infection. Yes, in this context, you will find also some publications which now found molecules that inhibit this so-called SEVI, this enhancer. So just to summarize, the science of our core facility is focused on antivirals, on antimicrobial peptides, and on anti-cancer peptides, where antimicrobial peptides include also antifungal peptides. And we have some international collaborations, for example, with the German University in Cairo, of course, with Professor Anselmo Otero from the University of Havana, recently also with Universidad de Rosario in Bogota, where we have got a grant from the Humboldt Foundation. And mostly these programs are related to exchange of students and exchange of master and PhD candidates. So could be for research stays for 2 or 12 months, for example. But sometimes these organizations like DAAD or Humboldt, they also fund some equipment or some materials. So in the context of your research consortium, very important to mention that we are working now very successful for 3 years in a project together with Anselmo, in a project which is funded by the BMBF about new antimicrobial peptides from invertebrates. As you know, Anselmo is from the Faculty of Biology, Havana University, and you may know this better than me. Snails produce exclusively peptides for natural defense, and therefore we have chosen these invertebrates as a quite good source to search for new antimicrobial peptides. And I think we have been quite successful, for example, in the case of CMP5, or also the peptides from Pomacea, the peptides from Nerita vesiculosus. So we have been in Cuba several times, have collected snails, have collected other sea anemones, other organisms, extracted the peptide fraction, and then screened on, for example, multi-drug resistant bacteria, problematic fungi, and of course also against new emerging viruses. So we have the possibility here to screen in security 3 laboratories. So the colleagues here are working with SARS-CoV-2 patient isolates. And as mentioned, together with the group of Anselmo Otero, we have more than 10 manuscripts in only, I think, only 3 or 4 years published or submitted. And as we have discussed before the seminar, the recent publication was about new antibacterial peptides from Pomacea. And we recently have applied for a 3 years prolongation of this project. And also we will participate in, or we have participated in an application of a so-called health center. This is a quite big grant application, which includes Cuba and I think also UNAM in Mexico City and further Ibero-American groups. The main applicant was Professor Vesuan from the Leibniz Institute in Halle. So to summarize our work here is, we can say that natural peptide libraries contain an abundant variety of different bioactive peptides. Screening for antiviral peptides resulted in a plenty of new antimicrobial peptides and different drug targets. And these natural peptides or their derivatives represent valuable molecules for pharmaceutical development. And we think that peptides and new biological entities in general will dominate the pharmacological market in the future. And with this last slide, I thank you very much for your attention. Hello?
Sorry,
Yes,
I have my
now I can hear you.
Off.
I can hear you.
Yeah. Some questions. The people are present have one question. Some questions.
I got a question.
Yes.
Yeah, please.
It's more like a comment more than a question.
Yes.
In fact, we've been working with antimicrobial peptides against tuberculosis and multi-drug resistant strains.
Yes.
But though the peptides have, some of them have antimicrobial activity, in fact, they have a good antimicrobial activity, but they also induce inflammation and actually a huge inflammation that the peptide kills the bacteria, but also kill the mice because of the huge inflammation. Most of them do this phenomena. So we've been trying to look for peptides that only have antimicrobial activity instead of the immunomodulatory activity. have you tried this in your peptides, or have you seen this same phenomena in your peptides, or how you choose or differentiate the mechanisms?
To be honest, most studies with our AMPs are still in vitro. So, but I remember during my Hannover time, we tried to develop an antimicrobial peptide, and when we injected it into mice, the mice were dying. So I think this is a very important point to consider, and yeah, maybe we should test earlier on such side effects to exclude such peptides, to work with peptides which later are toxic. Yeah.
And maybe you should to evaluate the immunomodulatory activities. Also, you can do it in a vitro model, maybe just evaluating chemotaxis or activation of dendritic cells. I don't know, but could be a good point for your further experiment.
Yes, of course. This seems a very important aspect. Yeah.
More questions? I take the opportunity to ask, in the case of peptides obtained from plasma, have you been able to work with plasma from infected patients, whether due to bacterial infection, fungi, or virus? Considering the fact that in many cases the synthesis of these peptides is stimulated by the presence of the pathogen.
Yes.
And then you increase the possibilities at the more specific, or you have more samples in the plasma.
Yes, you are completely right. We know, for example, many patients that suffer from chronic renal failure. They have diabetes, for example. And we also have collected, and they have an enhanced inflammatory status.
Okay.
And also, we have several patients here in, I don't know the word in English, they are treated by so-called cytosorb cartridges. So it's a life-threatening treatment because, for example, they have sepsis. And we also collect the sepsis filtrate, which is a cartridge which filters all the cytokines and all the acute phase proteins and peptides that are released in the body. This is also, yeah, of course, this is maybe a valuable source to start screening.
Okay. Thanks so much for other persons have the questions?
I have a question.
Yes, sure. Hi, Pablo.
Hi, how are you?
Hi, Tess.
Hello.
Hello. Do you evaluate the possibility to protect peptides from degradation, making chemical modifications or formulations in liposomes or such kind of things to protect the peptide in case they are injected?
Yes. In our group, we only can make peptide synthesis, for example, acetylation or amylation at the C-terminus. And yeah, but in our research center, we have other groups which are specifically dedicated to package peptides or to couple carriers to peptides, to put them into liposomes, to put them into silica, nanoparticles. So especially our work is only restricted to chemical modifications during peptide synthesis.
Okay, good, thank you.
Other question? People in the group, please.
May I ask a short question? This is very interesting, Professor. I was wondering if, in your experience, when you find these peptides from natural fluids, human fluids,
yes,
how common is it that you find like modified or particular amino acids that are not so common in comparison to regular sequences? Is there a balance in that?
What we often have seen are glycosylated peptides, which contain some short amounts of sugar chains. What we didn't often observe is strange amino acid residues or something like that. Of course, oxidation or things like that, but I think most PTM, post-transnational modifications, is glycosylation.
Okay.
Or también, also we have advanced glucated end products. We see also, due to the high sugar content of the diabetes patients, these patients carry peptides and proteins that contain sugar residues, which is very dangerous for the patients.
Okay, thank you.
More questions? About the service you have in your lab, the price is more or less how much you need to pay for one for synthesis or for identify use mass spectroscopy. The companies, for example.
To be honest,
Yeah, sure.
I'm more interested in research collaborations.
Is the first option?
Because we are here at the university, we have to generate some income, yeah. But our main aim is to make good publications, because with the good publications we can apply for a lot of money by grants.
Okay.
So, for my opinion, it makes not so much sense to have an income of 20,000 euro a year when we can have 500,000 of income making applicating for good grants. But of course, if you have a look at our homepage, you can see an official price list. Yeah, so you can order peptides, or we can make chromatographic fractionation for you. What we at the moment do not offer is mass spec. There is another, as mentioned, core facility which offers mass spectrometry.
Yeah, but
to give you an example, a multi-spectrum costs around 20 euro, or an LCMS maybe costs 100 euro. This is the range we are talking about. And the peptide synthesis, of course, if we have a common grant, we will not charge. Yeah.
Okay. Yeah.
If you order a peptide, a regular peptide, I would recommend you can order it in China. It will be cheaper. Yeah.
The collaboration is the good option for ours, sure.
Yes.
In this group is very big, and I am sure some people maybe in the next days have communication with you, because the options is very good for us.
Yes. No, I agree. Estoy muy contento que ustedes ya están trabajando con nuestros péptidos como Pomacea.
Sí.
Vamos a tener más
péptidos.
Péptidos nuevos, antimicrobianos.
No, excelente.
Yeah.
Bueno.
And it's always good to share ideas.
Perfect. I am sure you have maybe in the next days a lot of communication with some research from this group. Colegas, ¿alguna otra pregunta aprovechando esta oportunidad? Si no hay más preguntas, pues agradecerle mucho, agradecerle el tiempo. Es un trabajo muy bueno. Yo cuando empecé a acercarme al tema de los péptidos antimicrobianos, pues como que una de mis primeras ideas fue por qué buscar en animales, en otros animales, si el mismo cuerpo humano ya tiene las entidades, ¿no? que se han seleccionado y han defendido al cuerpo, y no pediría mayor diseño porque se aplican directamente en el sistema, están ahí. Pero la verdad es que las metodologías para poder obtenerlos de la sangre, por las cantidades en las que se encuentra, que son muy pequeñas, esa tecnología está a años luz de nosotros, ¿no? Todo el equipo que tú presentaste es algo con lo que yo siempre he soñado, y en los proyectos que escribo a gobierno federal para financiar el proyecto siempre incluye este tipo de cosas, pero son equipos muy, muy caros. Entonces, saber que hay un potencial, una potencial posibilidad de poder hacer uso de ellos mediante una colaboración o un servicio, es algo ideal para mí, y yo creo que para muchos grupos de la red. Bueno, pues nuevamente agradecerte tu tiempo, tu disponibilidad. Anselmo fue quien nos hizo favor de contactarnos. Él nos está escuchando por YouTube. Lamentablemente en Cuba no pueden entrar la conexión por Zoom. Y bueno, pues gracias, un placer conocerte.
Muchas gracias.
No sé si alguien quiere agregar algo.
Fue un placer conocer a ustedes y vamos a seguir en contacto.
Buenos días, doctor Carlos.
Sí, adelante.
¿Le habla Orlando?
Sí.
Hola, ¿qué tal?
Sí, tenía una pregunta. My English is not fluency.
Yes.
¿Puedo formular la pregunta en español? ¿No hay problema?
Sí.
Ok. De todo el trabajo que he visto que hacen, y hay algo que también un poco me tomó también un poco de preocupación cuando estuve en Bogotá. Quien le habla es de Perú, pero cuando estuve en una estancia en Bogotá, en el grupo de investigación en proteínas en la Universidad Nacional de Colombia, hice purificaciones de algunas fracciones de veneno de escorpión, y entonces un poco me preocupó era que cuando hice la evaluación antimicrobiana, había actividad, y cuando quise determinar la cuantificación y evidencia a través de electroforesis con la metodología de tinción nitrato de plata, no aparecía la banda.
Perdón, ¿no aparecía
la banda, en este caso,
la banda.
péptido, sí, el péptido teñido. Y entonces era para mí una cuestión, una pregunta interrogante: ¿a qué se debería, o muy poca la cantidad? Es posible, claro, y le hice, y entonces, claro, no teníamos un masas ahí, lo que teníamos en ese caso era hacer una electroforesis, y entonces era para mí un poco la interrogante, ¿no? Que había la evaluación antimicrobiana, en este caso se evidenció la acción antifúngica contra Candida albicans y sobre todo también contra Staphylococcus aureus, pero al momento de querer evidenciar, bueno, el péptido ahí o la fracción no se evidenciaba. Entonces, era ahí que se debería haber hecho en todo caso un masas de repente.
Sí. Bueno, si te queda algo de la fracción, se puede poner en una analítica HPLC o un mass spec, que son muy sensibles. En el STS page uno puede perder el péptido si es muy pequeño. Está fuera del rango.
Ok, muy bueno.
Si te queda material, se puede, a lo mejor se puede, con muy poca cantidad, se puede hacer un mass spec.
Ok, it's possible.
Sí,
So,
sí.
ok. Thank you very much. Muchas gracias.
Nada.
Bueno, pues de nuevo, le agradezco mucho tu tiempo y gracias a todos por participar en este seminario. Fue el último seminario del año, nos fue este año tan particular, se nos fue muy rápido, tan particular y tan difícil. Y bueno, pues nos vemos el 100 seminarios en enero, el segundo viernes de enero, porque creo que el primer viernes de enero es creo que primero o segundo o día 2, no me acuerdo. Entonces lo dejamos para el segundo viernes. Y ahí les mando la invitación y el recordatorio del grupo que les corresponde. Doctor, muchas gracias por su participación y estamos en contacto, seguramente.
Ok,
Sobre todo
gracias.
sobre todo por POM 1.
Yes, of course. Hasta luego.
Hasta luego, mucho gusto. Hasta luego a todos, gusto en saludarles, síganse cuidando.
Bye.
Bye,
Que estén
bye.
muy bien.
Hasta luego.
Gracias, hasta luego.
Hasta luego, gracias por
Luego.
conectarse.
Luego.
Bye, bye.